Hi, I'm Andrew Penn. I'm an associate clinical professor in the UCSF School of Nursing. And today we're going to be talking about basics of receptor pharmacology. Receptor pharmacology refers to pharmacodynamics, which is what the drug does to the body. This is a complex, fascinating, and engaging topic, which you may find that you need to go over more than once, but I'm going to try and break it down into some digestible chunks. So today we're going to be talking about the key components of neurotransmission, including the synthesis and release of neurotransmitters, where those neurotransmitters attach to, and what breaks them down. We're also going to talk a little bit about what the neurotransmitter does at the receptor and to be able to identify what some of these key neurotransmitter systems are and what they're responsible for doing. So the idea of neurotransmission is a relatively, in the whole history of the world, relatively new discovery. It goes back into the 19th century. I want to contrast two neuroscientists from two different centuries, Santiago Ramón y Cajal, who is a Spanish neuroanatomist who did beautiful drawings of the nervous system under the microscope in the early 20th century, and a gentleman by the name of Robin Carhart Harris, who's doing fascinating functional connectivity MRI work with psychedelic drugs such as psilocybin in the UK. And while these two may be a century apart, they are describing aspects of this very complex but very important portion of the human body. So we'll talk about what these different actions of drugs are, what is a neurotransmitter, and what does it do when it gets to the other side of the synapse in the most basic fundamental terms today. And I want to underscore that this is some complicated stuff. This may be some of the most complicated material in the entire course. And what I've illustrated here is if you've spent any time hiking around Northern California, you've undoubtedly seen these big black turkey vultures that like to fly in big arcs over the hillsides of Northern California. And I think about these birds because what they do is they follow a thermal of hot air going up into the air. And I think, gee, when that bird looks down at the ground, and it thinks, my gosh, I'm over that same barn that I went over just a few minutes ago. I must not be going anywhere. But what happens with each subsequent pass is it goes a little bit higher. And I imagine as it goes higher and higher still, it looks down and it starts to see how things are connected. It says, wow, I didn't realize that the ocean was right over that mountain there. And that road actually connects to the next town over. And so that is similar to how learning complex material, I often think of it as this kind of iterative process of going over the same material over and over again. And with each successive pass, you learn a little more. And so it is with psychopharmacology. It can be very daunting when you first begin to learn it. But what you'll find as time goes on is that the information starts to become more coherent and you start connecting the dots. So we talked before about pharmacokinetics, which is essentially what the body does to the drug. That's where the body absorbs the drug, it breaks down the drug, it distributes it around whichever organ system or throughout the bloodstream. Hopefully some of it goes to its target organ, and then eventually how does it leave the body? So that's pharmacokinetics. What we're going to be talking about now is pharmacodynamics, which is how the drug affects the body. And particularly when we're talking about drugs that are used in psychiatry, we're generally focusing on the brain and the central and peripheral nervous system, of course. Now, there are over 100 million neurons in the brain. And as you remember from your anatomy course, these neurons are connected to one another, they don't actually touch. There's this very tiny little gap called a synapse, and there are over 100 trillion connections between these neurons. So this is the most complicated supercomputer you can imagine. And what we're doing with most of our medications is we're altering the communication between the neurons. And so if that is an immediate effect, you can affect, say, for example, whether or not that neuron fires. Or we can have later downstream effects, which can cause, say, for example, the neuron to grow in response to an antidepressant treatment. But that is a slower process, what we often refer to as a downstream effect. And it's also important to remember that even though we can make these changes, the organism often will respond to these perturbations, especially if they are chronic perturbations, by these kind of compensatory changes at the synaptic cellular level. So a good example of that you're probably familiar with is tolerance to opiates. So when somebody takes an opiate chronically, they become less sensitive to its effects because of the adaptations that the cells make in response to that chronic exposure to the opiate. We'll talk more about that as we go along. So you're probably familiar with a diagram like this. I just want to walk you through this schematic. This is an example of a synapse, and you can see I've blown up that little section on the left to highlight the smaller features of the synapse. Let's go through them one by one. So first of all, when the signal comes from the presynaptic neuron, it's flowing downstream from top to bottom, if you will, in this diagram. So when that signal arrives and begins to depolarize the membrane at the synapse, a number of things can happen. One of them is that the synaptic vesicles will open up. In fact, the cell wall opens, and these neurotransmitters, which are symbolized by these small blue triangles, begin to drift across the synaptic cleft or the synaptic gap. And their target are these receptors on the other side. I mean, really, they're just kind of floating around, but where they will be received is in those dendritic receptors on the postsynaptic neuron. Now, the human body likes to be efficient, and so it's not going to allow those neurotransmitters just to go to waste. So it uses something called a reuptake pump or reuptake transporter in order to pull some of those neurotransmitters back into the presynaptic terminal to reuse them. It's essentially like a recycling mechanism. Also, within the presynaptic neuron, there are these vesicular transporters that reload those vesicles so that they are primed and ready to go. They're already packaged up and ready for release. There are enzymes in the synaptic gap, such as monoamine oxidase, which break down the neurotransmitters, such as this little Pac-Man is symbolizing here. So what are some common neurotransmitters? Well, some you've heard of: acetylcholine, glutamate, GABA, serotonin, norepinephrine, melatonin, dopamine, anandamide, and orexin. These are just a few. There's others such as histamine that you've also likely heard of, but not maybe realized that they were neurotransmitters. What is a neurotransmitter? Well, what it is, it's really this chemical substance that is stored, as I showed you, in the presynaptic vesicle, ready to be released when that nerve terminal is depolarized. And it binds over to those receptors on the other side. And there are some exceptions to this, something called retrograde signaling, which we'll get into when we talk about endocannabinoids. And once it plugs into the receptor on the other side, it elicits usually a fairly quick response, which can be reversed. And then eventually that neurotransmitter is either going to diffuse away, it's going to be released from the receptor, it's going to float away, or it's going to be broken down by one of those enzymes like I showed you in the example of monoamine oxidase. Or it will be pulled back up into the presynaptic terminal by the reuptake pumps. So that's what characterizes a neurotransmitter. And what's a receptor? So a receptor is just simply usually a protein that is on the surface, and most commonly it actually sort of sits it bridges the membrane of that cell. So it's what we sometimes call a transmembrane protein or transmembrane receptor. And so that means that part of the receptor is sticking out of the cell and part of it is inside the cell. And these receptors are specific targets for neurotransmitters. And when neurotransmitters are made by the body, we refer to them as endogenous. And often the term for what plugs into a receptor, kind of like a plug that goes into a socket, is a ligand, or if you're from other parts of the country, they call it a ligand. And that's really a chemical messenger that can be made by the body, or it can be a drug. When it's a drug, we refer to it as exogenous, meaning it comes from outside the body. And once those receptors are attached to by a neurotransmitter or a drug, then there is a downstream effect that happens to that receptor. And we'll talk a lot about that as we go on through this course. And that receptor, there's a range of things that can happen to that receptor. It can either be activated, which means that it's turned on. So a process that receptor is responsible for begins to be activated, much like a light switch, or it can be held in a neutral position. So this is similar to like when we, if you have a toddler in the house and you've put those little covers over the light sockets, or electrical sockets rather, it prevents that plug from being activated. Or it can even cause the opposite activity of whatever that receptor does, which is what we call inverse agonism. We'll talk more about this in the next module. There are also more subtle effects that can be confirmed by a drug, or sometimes a neurotransmitter, called allosteric modulation. So these are things that enhance the effect of another ligand. So these can either increase the activity at the receptor, or it can decrease the activity at the receptor. So that's positive and negative allosteric modulation, respectively. And this will be important to remember when we get to benzodiazepines, things like lorazepam or diazepam. Here's a visual of what that allosteric modulation looks like. You can see that top cylindrical object is the agonist. That is the neurotransmitter plugging into that key-like shape in the purple receptor. Now, if a allosteric activator is also plugged in to that triangular shape, these almost look like those kids shape sorting toys, the activity of that receptor will be increased, as you can see in that schematic in the top right corner. So the maximum output you could get is a agonist plus an allosteric activator, A plus C. If there's an allosteric inhibitor, as there is with A plus D, it still turns that switch on, but it turns it on at a much lower level. So you might think of this as sort of like a dimmer switch. It's the light is still on, but it's much less bright than it could be. So this is allosteric modulation, both positive and negative. And where the rubber really meets the road with regards to pharmacology is how different drugs work on different parts of this system. And this is, we're going to spend a lot of detail on this in the weeks ahead, but I just want to give you a 30,000 foot view and a few kind of prototypic examples of where different drugs work. So for example, a drug may be a neurotransmitter releaser. A classic example of this would be amphetamine, which releases dopamine and norepinephrine. Another drug called methylene dioxymethamphetamine, or MDMA, causes a release of serotonin from the vesicles. So that's one way that a drug can work, is just by pushing more neurotransmitter out into the synaptic space, so it's way more likely to make it to the other side of those receptors. You can also have partial agonists, or a full agonist. So this is where a drug acts like a mimic of the endogenous ligand or the endogenous neurotransmitter. So it's working, so for example, a morphine molecule on a mu receptor will activate the receptor in the same way that the endogenous ligand is activating it. And if it's only activating it partially, it's what we refer to as a partial agonist. But this is what agonism is in a receptor. You can also block that receptor in the same way that I mentioned before you could do with a cover of an outlet. So haloperidol is a dopamine receptor antagonist. It blocks dopamine from docking into that receptor site. You can also have something called autoreceptors. Now, autoreceptors, you'll notice, are up here on the top center of your screen, these are on the presynaptic neuron, and they work sort of like a thermostat. So they are a target for the neurotransmitter that's being released, say in this case, norepinephrine. And the idea is that once enough norepinephrine is released, some of that's going to float up to that autoreceptor and plug in. And just like a room gets up to a certain temperature and then it tells the thermostat tells the heater, you can go ahead and stop making heat now. This is how an autoreceptor works. So it essentially is a feedback loop which tells the presynaptic terminal to go ahead and stop releasing those neurotransmitters. So mirtazapine or Remeron is a drug that causes the autoreceptor for norepinephrine to be blocked. You can also have reuptake pump inhibition. This is a very common way that antidepressants work. So it causes that reuptake pump, that recycling mechanism that we talked about, to be slowed down or inhibited. And by doing that, we allow the neurotransmitters to stay in the synapse for a longer period of time, so they are more likely to make it to their receptor targets. You can also inhibit the enzyme which breaks down those neurotransmitters. So the oldest antidepressants are called MAOIs, or monoamine oxidase inhibitors, and they prevent the breakdown. They prevent the enzyme which breaks down serotonin, norepinephrine, dopamine, which are the monoamines, prevents those from being broken down by that MAO inhibitor, that MAO enzyme. And then finally, a newer group of drugs called VMAT inhibitors. They prevent the reloading of that presynaptic vesicle. So some of the newer drugs that have come out in recent years to address tardive dyskinesia are examples of VMAT inhibitors. So in summary, what we're doing is we're affecting the way that neurons communicate with each other, or neurotransmission. And there's many different ways that we can do that, usually by manipulating either the receptor or the release of the neurotransmitter. But there's many different and subtle ways that we can make changes in this, and there are some not so subtle ways that we can do that. But that is really at the heart of what psychopharmacology is all about, is manipulating these different communication pathways. And we'll get more into detail about which neurotransmitters we're manipulating and with what drugs in the future modules. Thanks.